you guys know, my name is Banister. I'm here today to talk to you about genome sequencing for the purpose of phylogenetic relationships. That wasn't the bad button. I didn't press the bad button there. I don't know what's going on.
Maybe it got pressed before. Okay.
What?
Press it again. Press the bad button again.
I'm not pressing.
I don't know. Use the space bar.
All right, sounds good.
I'm sorry. You just have to use that for the pointer.
Okay. Time restart?
Yeah, I'll take off 45 seconds. Go ahead.
Okay. The topics I'll be talking to you today about is genome sequencing, storing of information in computers, and creating phylogenetic relationships. Now you might ask, why is this important? Why do we need to know about this? It helps to understand papers. So if you're doing a paper on infectious diseases or some type of evolutionary system, this comes from genome sequencing. That's the most basic form. It's all the way down to the basic nucleotides that make up DNA. It can also prevent studies and prevent optimal results if you don't know about this, this process. If you're doing a method that's outdated or that doesn't give you the best results, and you're not aware of the new technology advancements in genome sequencing, then you won't be getting the best results that you need for your study. Now, quick review. I have this photo right here. It's the traits that come from genes, as you can see right here. This is a genome sequence that makes up the red pigment. These genes are made up of adenine, thymine, guanine, and cytosine. And then phylogenetics. Phylogenetics is the comparison between species of its physical traits related to its genes. So you can see here in the picture, the red pigment is in all of these species right here. And this is the trait that makes it up. So this helps with comparing these genes and seeing where these genes or traits appeared in evolution. Genome sequencing has been at a high for the past 20 years. It's basically due to technological advancements. It's heavily used by computers. That's heavily computerized. So that's why we see the growth that we see today. It's very valuable to scientists, different companies, and health organizations. It's really exploded during the Human Genome Project, which was done by 20 different institutions over 6 countries. And their goal is to complete the sequence of the human genome. They use 2 different processes called the whole genome shotgun approach and the hierarchical approach. The hierarchical approach involves 3 different stages. The first stage is linkage mapping, which deals with the denaturing of DNA strands. This is done in 4 different types of test tubes. Each test tube has identical template strand within each. It has a DNA polymerase and a primer. Where the differences come within these test tubes is the 4 dideoxyribonucleotides. These are fluorescent indicators that attach to the DNA strand and cut it into fragments, which leads to the second step of the physical mapping. Physical mapping is the cutting of specific gene fragments, and once this is done, it can be sequenced, which is the third step, by a fluorescent indicator, which reads the fluorescent indicators attached to the genes. Now right here I have a picture of a DNA strand. This blue dot right here, it symbolizes the fluorescent indicator that attaches to the DNA strand in the test tube, and it prohibits from any further production of DNA sequencing going on. So when you combine them all, this is one strand here, and you can see that it has the same sequence going up, and at the end of each has a different color. These are the fluorescent indicators that cut it into the different strands. Once this is done, it can be sent through a fluorescent detector and then sequenced into a spectrogram, which can be then analyzed and compared. The whole genome shotgun approach is a faster method. It was developed by Craig Venter in 1992, and it skips the whole process of linkage mapping, which was what was done in the test tubes with the 4 different fluorescent indicators. It's cut, cloned, and sequenced completely by computers. You can see in this picture here, this test tube here includes the full DNA strand, and it's only one, and it's inserted into the computer, and it's sent through the system at a speed which breaks up the DNA sequence randomly. So there's no particular spot where it breaks. It just sends it, the speed breaks it into just random pieces. This is where it gets its fault though. It misses random sequences because sometimes the speed isn't fast enough to break the DNA strand small enough, so it becomes hard to sequence. This is a very fast method though. It was completed the human genome 2 years prior than the Human Genome Project expected. But both methods are still exist today. Because this fault, this method has a fault of missing sequences. The last one takes a lot of time, and time is money. After sequencing the human genome, they've sequenced over 600 genomes. As of 2007, this number was expected to double every 10 months. So you can imagine how many genomes have been sequenced by today. This includes many archaeal species, eukaryotic, and bacterial species. It's mostly bacterial species that are sequenced because they tend to have a smaller genome, so it can be done a lot quicker, a lot cheaper. It also tells us a lot about infectious diseases that relate to humans. Now, where is this information stored? The human genome itself includes approximately 3 billion base pairs. That's a lot of information, and when you start getting to over 600 species being analyzed, it's difficult to find an area to store all this information. So that's where the new field of bioinformatics is created. Bioinformatics is basic biology information, and this deals with genes, genomic sequencing. Now, this is stored all in GenBank. GenBank includes a series of websites, including the National Institute of Health and the National Center of Bioinformatic Information. These are Human Genome Project and government-funded websites. And it makes it available to the world. So as long as you have an internet connection, you can look up these comparisons and this information on the computer. Now, this information holds clues to evolution. For example, when a new trait or chromosome is formed throughout evolution. It also compares related species, which is the process of phylogenetics, as we discussed earlier. Now, large advancement in computer technologies. This includes the overall better computer programs. As it's advanced, people have developed better computer programs to compare these traits. The speed of these programs has increased, and the range of DNA substitution programs have increased as well. Evolution application is based off likelihood, so it's the likelihood of comparing one sequence to another. So if you have a series of species, you can compare their genes and see where different mutations occurred, and by mutation you can see where these genes or where these species appeared in evolution. The two types of computational processes are likelihood ratio testing and the maximum likelihood estimates. Likelihood ratio testing is normally used for finding comparisons between specific genes. So, for example, like the red pigment gene that we looked at earlier. These models and in the computer have specific parameters set to them, so they're set out to locate the specific trait and compare species based on that trait. Now, maximum likelihood estimates is more common. It covers a wider variety of species, and it includes 3 major parameters of speed, amount, and unusual behavior of mutations. But when you insert too many of these parameters, like too many of these assumptions, it results in a bad model. This is because you're controlling the information too much. The likelihood of, or the calculations don't have the freedom to work and compare with one another vastly, so this will result in a bad model. Now, these maps that I've been talking about, these models, are seen right here. Many of you have seen these before. They're called dendrograms. These show the evolutionary process to get these species right here now. And it gives an understanding of how these species evolved. So different sites of where these mutations occurred. Its biggest purpose that we see today is for trends related to infectious diseases. Health is a big topic, and today people are concerned about human health. So this is why we see most of this related to the infectious diseases. Phylogenetic branches are these branches you see here, the blue lines. Lots of branches mean many mutations. So this here would have a series of mutations, which would be more than, say, just right here. Now, the longer the branches means a larger change within the species. So this species A is going to be a lot different than species B. And that's determined, or you can be seen through the length of the branches which are separated. The models are based off calculations. Calculations are based off the likelihood of these genes and their relations, and likelihood is based off the genetic makeup. Designated ancestral species, normally they'll be inserted prior to sequencing or creating phylogenetics, but sometimes they can be misinterpreted. Sometimes they could believe that an ancestral or one species is the ancestral species, but they could be wrong. So these models can be reversed within the program. It'll set each species as the ancestral species and recalculate, and this way they can come up with the most likely ancestral species and cover more bases. Now, models within studies are labeled specifically, so these are examples of the HKY model, FAI, and JC. When you're reading a research paper, you'll see that they say use the HKY model. Now, this is just an example. The HKY model is very large, but say the HKY model just includes these. Now, the JC model might include all of these, and then the FAI could be a full larger process. The increase in freedom that your information has, the better ability the programs have to direct, calculate, and interpret the information, but the less parameters means it overlaps with hypotheses. Now this means that there's too many ancestral species, which means that there's a lot of unneeded data that you need. So you don't need to be comparing sometimes a species that is generally, for the most part, not related to another. If you have too many of these, it just becomes too vast, and there's just unneeded calculations that are being done. Now, this is where the null hypothesis is introduced. This is a hypothesis in science that is on when your data won't be changed by adding or removing different parameters. Now, that's one of the goals of phylogenetics, is finding a point in your data when inserting a different parameter in your program will not change the data or data resolving. Less variation of the species means more dependable data. So if you're looking at specifically these two, you're going to be able to analyze them a lot thoroughly and a lot better than analyzing a wider variety of species. Now, two dendrograms usually result. This means that when you're done these calculations and the computer, a dendrogram, say that includes A, D, and C right here, will resolve, but there will be two of them. So one will have ADC arrangement like this, but the other will have the A switched with the C. Now this happens just because traits, they end up being, the larger traits outweigh the defining traits. So you have a large trait within your sequence, but it will outweigh the defining traits that actually define the difference between the species and the mutations in the species. Now, bad models are a result of underestimating the species involved, which includes the mutation rates. A lot of the time, they're unaware of how quickly these species evolved and how many of these species evolved.
You must have hit the button as you.
Oh, no, yeah, so this is from overestimating the species involved. So, to conclude, the genome sequencing is a result of rapid technological advancements. That's why you've seen its growth in the past 20 years. Sequencing has improved because of technological advancements. Calculations have improved because of better programming, and phylogenetics has improved because of a vast variety of studies. Now, it holds a lot of great potential and answers a lot of questions like what makes us human, what infectious diseases affect humans, and where they come from. And it's beneficial to different businesses, scientists, and health organizations. For an example of it being beneficial to businesses, say you're in the food industry and your product is beef. We all know about the mad cow disease that came up a while ago. This is a result of a mutation that occurred within the DNA sequence, and this disease appeared in cattle. As a business, you don't want to be obviously infecting people with diseases from your product. So this is when health organizations and scientists become involved. They can go in and see where this mutation occurred, how it occurred, and when it occurred, and hopefully they can figure out an antibiotic or a way to prevent this from affecting humans. Now, some drawbacks to this: it's a very expensive process. So you have to be able to prove why you deserve to be funded, why your research deserves to be funded. And to do this, you have to be able to prove the size of your genome. The bigger the genome, the more money it's going to cost. The more time you need, the more people you need to pay. Is phylogenetic relation to humans? People care about humans. This is the way we are. Businesses want to know how it's going to affect humans. So this is an important process or thing to define to the business to get funding, is how it affects humans. And if it provides further research, that's also good. If you can go back to the research and continue doing further studies, that's very beneficial.